Hey everyone, welcome to today's podcast. Let's talk about something fascinating and super useful in the world of science and medicine, immunohistochemistry, or IHC for short. Sounds fancy, right? But trust me, it's not as intimidating as it seems. So grab your coffee, sit back, and let's unpack this step by step. What exactly is immunohistochemistry? Alright, so IHC is this amazing lab technique where scientists use antibodies, those little protein warriors in our immune system, to hunt down specific antigens, think, unique proteins, in tissue sections. Why is that cool? Well, it lets us actually see where these proteins are hanging out in cells, which is a game changer for diagnosing diseases, predicting outcomes, and even advancing research. Yep, you heard that right, tiny proteins, big implications. Breaking down the steps of IHC, let's walk through how this works, step by step. Imagine you're in a lab, and you've got this tissue sample you want to analyze. Here's what happens next. Step 1, tissue prep first. We've got to preserve the tissue. Usually, this means dunking it in formalin to keep everything intact. Then, we embed it in paraffin wax, like making a candle, but for science, or freeze it for sectioning. Step 2, slice it thin. Next, we cut the tissue into super thin slices, about 3 to 5 micrometers. That's thinner than a strand of hair. These slices are prepped for the magic to come. Step 3, deparaffinization and antigen retrieval. If you're working with paraffin-embedded samples, you've got to remove that wax with chemicals like xylene. Then comes a crucial step, antigen retrieval. This is where we use heat or enzymes to expose the hidden epitopes, or binding sites, on the antigens. Think of it like unlocking a treasure chest so the antibodies can do their job. Step 4, blocking. Blocking is next. We use agents like serum or BSA, bovine serum albumin, to prevent non-specific binding, basically, to make sure the antibodies don't stick where they are not supposed to. Step 5, primary antibody incubation. Here's where the antibodies come in. The primary antibody is like the detective, it's designed to specifically recognize and bind to the target antigen. Step 6, secondary antibody and detection. Now, the secondary antibody binds to the primary one. This secondary antibody is labeled with an enzyme, like HRP or AP, or a fluorescent dye, which helps us visualize the target antigen. It's like shining a spotlight on the culprit. Step 7, chromogen application and counterstaining. Here's where it gets colorful. A chromogen reacts with the enzyme to create a visible signal. Brown staining with DAB is pretty common. Then, we use a counterstain like hematoxylin to highlight the cell nuclei, adding contrast for easier viewing. Step 8, microscopic analysis. Finally, it's time to look at the stained tissue under a microscope. This is where pathologists examine the patterns and intensity of the staining to reach a diagnosis or insight. Why do we use IHC? Now you might be wondering, what's the point of all this? IHC is like the Swiss Army knife of diagnostics. Here are some of its key uses. Cancer diagnosis and subtyping. For instance, it helps us identify tumor origins, things like cytokeratin for carcinomas or vimentin for sarcomas. It's also used to detect hormone receptors like estrogen and progesterone in breast cancer. Prognostic and predictive markers. Markers like HER2/NU guide treatments for breast cancer. Hello, trastuzumab, while Ki-67 shows cell proliferation rates. Infectious diseases. We can detect viral proteins like those from EBV or HPV and even bacterial antigens. Autoimmune and neurological disorders. Think lupus or Alzheimer's. Imagine catching immune complexes or amyloid plaques with precision. Research. And don't forget the lab. Researchers love IHC for studying proteins in cell signaling, drug development, and way more. Advantages and challenges. Of course, no technique is perfect. Let's quickly break down the pros and cons. Advantages: high specificity. These antibodies know exactly what they are looking for. Localization. It shows precisely where proteins are in the tissue. Multiplexing. Detect multiple markers at once with advanced methods. Clinical utility. It's a decision maker, especially with therapies like PD-L1 for immunotherapy. Challenges: variable sensitivity. Sometimes antigens are masked and hard to access. Cross-reactivity. Antibodies can bind to the wrong stuff, causing false positives. Subjectivity. Scoring. Staining intensity can depend on the observer. Cost and time. Antibodies and reagents don't come cheap, and the process can take a while. Known antigens only. Yep, No surprises here, it works best if you know what you're looking for. So, there you have it, immunohistochemistry in a nutshell. It's a technique that combines the precision of science with the art of visualization, making it indispensable in modern diagnostics and research. Thanks for tuning in today. If you found this helpful, don't forget to like, share, and subscribe for more deep dives into the fascinating world of surgical pathology. See you next time. Everything Surgery Podcast. Dissect the extraordinary.